We usually think of evolution as a slow process over millions of years, but for bacteria, adaptation is an active strategy playing out right now. This is the Harvard megaplate experiment. The outer edges have zero drugs, but moving inward across vertical boundaries, antibiotic concentration increases exponentially to 1,000 times lethal dose. Bacteria grow, hit a chemical wall, mutate, and break through in just 11 days. This localized, rapid adaptation stands in contrast to the classical model of eukaryotic evolution, which is characterized by slow, vertical descent across millennia. To understand the emergence of modern superbugs, we must abandon our traditional mammalian timelines. We need to examine an evolutionary engine operating at a hyper-accelerated scale. The foundation for observing this continuous evolutionary math is Richard Lenski's long-term evolution experiment at Michigan State University. This schematic details the study's methodology. Researchers isolate specific bacterial populations and perform daily serial transfers into fresh media, tracking their genetic changes continuously over decades. The experiment highlights the first mathematical driver of bacterial evolution: extreme replication speeds. Under optimal conditions, a species like E. coli has a generation time of roughly 20 minutes. This graph shows an exponential growth curve, illustrating our second driver: massive population density. A single milliliter of liquid culture grown overnight can contain up to 10 billion individual cells. This massive numerical canvas provides the exact statistical probability required to fuel rapid genomic variation. These extreme replication speeds and population densities fuel a three-part engine of genomic variation: point mutations, genomic rearrangements, and horizontal gene transfer. We begin at the microscale with point mutations, specifically single nucleotide polymorphisms, or SNPs. A SNP is a single base pair change in the DNA sequence that arises due to inevitable errors during replication. These base pair errors take two forms. A transition occurs when a purine is swapped for another purine, or a pyrimidine for a pyrimidine. A transversion is a more drastic structural swap between a purine and a pyrimidine. Because multiple codons can code for the same amino acid, these swaps often result in a silent mutation. The underlying DNA sequence changes, but the resulting protein remains exactly the same. If the SNP alters the final amino acid, it creates a missense mutation. This structural change can provide a novel physical benefit to the resulting protein. Alternatively, the error can create a nonsense mutation. This introduces a premature stop codon, abruptly halting translation, and generally resulting in a non-functional truncated protein. Beyond single substitutions, replication errors also introduce insertions and deletions, known as indels. An indel involves the physical addition or removal of entire nucleotides from the sequence. Indels carry a severe structural impact because they cause a frame shift. By misaligning the three-letter reading frame, an indel scrambles every single downstream codon, entirely altering the protein architecture. Most of these mutations are neutral or lethal, but with 10 billion cells replicating every 20 minutes, a rare, highly beneficial missense mutation is statistically guaranteed to occur. Under selective pressure, that single variant will survive and sweep the entire population. Scaling up, the second driver is genomic rearrangements. These are larger architectural shifts within the organism's existing DNA. One key rearrangement is gene duplication. It acts as an evolutionary sandbox, allowing bacteria to experiment without risking the original gene's survival function. Over generations, the original copy continues performing its vital cellular function, while the duplicated copy is free to safely accumulate point mutations. Eventually, that mutated copy diverges entirely, becoming a distinct functional sequence, providing the organism with a novel trait. A distinctly different type of rearrangement is an inversion. Bacteria utilize inversions for phase variation, a process of rapid, reversible adaptation. We can observe this mechanism in the FIM-A gene of E. coli, which controls the expression of fimbriae, hair-like appendages used to attach to host cells. Here, the central promoter sequence is flanked by two inverted repeats. During an inversion, the promoter region physically detaches, flips 180 degrees, and locks back into place. This breaks the reading frame, acting as a mechanical on-off switch for fimbriae expression. By leveraging these genomic rearrangements, bacteria maintain the architectural flexibility to drastically alter their physical phenotype, allowing them to evade a host's immune system. The third and most aggressive driver bypasses vertical evolution entirely. Horizontal gene transfer, or HGT, is the ability to acquire massive data packets of completely foreign genetic material from neighboring organisms. There are three main pathways for HGT. The first is transformation, the passive uptake of naked, free-floating DNA fragments from the surrounding environment. The second pathway is transduction. This occurs when a bacteriophage virus acts as a biological syringe, injecting a payload of foreign genetic material directly into the cell. The most potent pathway is conjugation. This is the active, deliberate transfer of circular DNA structures called plasmids between two living cells. To facilitate this transfer, the donor cell extends a physical bridge, known as a sex pilus, to connect directly with the recipient. Conjugation enables the transfer of entire pathogenicity islands, dense clusters of genes that encode complex, coordinated biological weapon systems. For example, the PMAR conjugative plasmid encodes a complex bundle-forming pilus required for specific strains of E. coli to initiate severe intestinal infections. An even more extreme example is the PUUH239.2 plasmid, notorious for driving severe, untreatable Klebsiella pneumonia outbreaks in hospitals. That single transferable package carries over 13 distinct antibiotic resistance genes. Horizontal gene transfer shatters the biological speed limit. It allows bacteria to instantly download complex survival toolkits without waiting for the slow process of random mutation. This brings us to a foundational taxonomic problem. If bacteria are constantly mutating, rearranging their architecture, and swapping massive gene packets, how can classical biology define a bacterial species? These Venn diagrams illustrate the pangenome paradigm. The central core genome contains the essential sequences required for the baseline survival of every strain. Surrounding that is the accessory genome, a highly fluid shell of specialized tools acquired via horizontal gene transfer. Under this paradigm, a bacterial species functions as a dynamic network, constantly shifting its architectural boundaries. Highly specialized, isolated bacteria that rarely share genes have what is called a closed pangenome, but the pathogens that threaten human health thrive on a highly fluid, open pangenome. E. coli is the ultimate example of a species utilizing this open pangenome strategy. This constant flow of accessory genes dictates the organism's pathogenicity. It is why one strain of E. coli can be a harmless resident of your gut, while an EHEC strain can cause a lethal infection. The difference between them is housed entirely within that acquired accessory genome. When we look back at the Harvard megaplate experiment, the true threat becomes clear. Antibiotic resistance isn't born in isolated pockets. It's rapidly shared, accumulated, and refined across populations. Their open, modular evolution makes them the ultimate biological survivors. Operating on a hyper-accelerated timeline, they represent the greatest ongoing challenge to modern medicine.